All right, so like I said, we're going to go ahead and get started with our chemical monitoring workshop and start out by kind of going through and telling you a little bit about Georgia Adopt a Stream. So Georgia Adopt a Stream is the state's volunteer water quality monitoring program that trains citizens to go out and monitor their local waterways, their streams, lakes, wetlands, estuaries, coastal, whatever it might be, whatever you're interested in learning more about. And we are housed in the non-point source unit of the Environmental Protection Division of the Department of Natural Resources. So a big mouthful, but all that is to say that we are with the state of Georgia. And so we have kind of this these program goals that are laid out in this handy dandy mnemonic adopt. And for everyone that is here looking to get their certification, I would pay special attention to everything that is in bold, that is on our slides, because that may or may not, aka 100% will appear on your certification test that you'll take at the end of your field portion. So again, getting started with the program goals. So first up, we have A, that stands for awareness. So we are striving to increase public awareness of water quality issues in the state of Georgia, and specifically non-point source pollution, which is the biggest threat to water quality in throughout the state. And we look to increase awareness by conducting workshops, hosting outreach events, river cleanups, discussing our monitoring efforts, and so much more. And in general, people don't understand their impacts on their local waterways. And so we hope by getting people involved in our program, then they can learn a little bit more about how to mitigate their impacts. Next up, we have data. So our volunteers collect baseline water quality data according to Adopt a Stream protocols. So when our volunteers go out and they take their samples and they submit their data, they submit it to our online database, which is open access, so anyone can look at the data. The program's been going on since 1993. So when they're submitting their data onto our database, they're submitting it into this really long-term widespread data set that is super helpful for understanding kind of the baseline conditions of our waterways across the state. And this is, again, incredibly important because a single, set of scientists or even a regulatory agency is not going to be able to collect data on every single waterway across the state super consistently. And so by having our volunteers go out and do that, it's really, really helpful for understanding kind of what's normal and what's the baseline conditions for our waterways. Next up, we have observations. So every single time that our volunteers go out and collect data, regardless of which monitoring program they are involved in, they always take observations of the site and note water quality conditions. So those observations can be things like water color, water clarity, maybe whatever's on the surface, if there's any sort of oil or if there's algae, whatever it might be. These observations are incredibly helpful for kind of understanding what's happening in your waterway and also putting your data into context. So for example, if you're seeing really, really low oxygen levels in your stream and you see a whole lot of, nasty decaying organic matter, then that can help you understand a little bit about what's happening in your data. Next up, we have partnerships. So we definitely emphasize our volunteers creating partnerships and forming partnerships with their local governments, with local nonprofits and other organizations, because not only do these relationships help with regards to equipment and helping guide your monitoring efforts, but you can also, it helps you understand who to call and who to contact if you are seeing any pollutants or anything that's harmful entering your waterway. So it really helps working together to solve these problems in our waterways. And finally, we have T, which stands for tools and training. So that's kind of what we're doing here tonight. We are providing training for you all in the chemical monitoring program. And additionally, we have our local coordinators across the state who can serve as a point of contact and help guide you in your monitoring efforts, and also possibly provide monitoring equipment for you to use when you go out and sample your stream. All right, so now we're going to talk a little bit about the types of pollution that you can see in your stream. So pollutants generally are substances that, when introduced into a system, can have adverse effects on the surrounding environment. And there are kind of two broad types of pollution that we see in our waterways. So first up, we have point source pollution, which is really easy to remember because you can think of it as you can point to the source of pollution. It's really easily identifiable. It can be something like, you know, a pipe like you can see here, like a construction site, a wastewater treatment plant, anything that's very obvious as to where the source of the pollution is. And point source pollution in general is very well regulated in the state of Georgia and in the US in general due to the Clean Water Act. And so the Environmental Protection Division of Georgia has a point source regulation permitting process essentially that's called the NPDES process, or National Pollutant Discharge Elimination System. So again, point source pollution is definitely pretty well regulated. But on the other hand, there's also non-point source pollution, which is the exact opposite of point source pollution. You can't really point to the source. The sources aren't really able to be distinguished or identified. And essentially everyone is contributing to this pollution. So anything that is happening on the land that people are doing can have an impact on our waterways. So things like fertilizer on an agricultural field or on a lawn, people not picking up after their pets. And so that E. coli is getting into the streams. Anything like that is an example of non-point source pollution. Because everyone's contributing and because it's much harder to regulate, it is by far the #1 cause of water quality problems in Georgia. And it is what we are focused on in the Adopt a Stream program. All right, so now we're going to talk a little bit about watersheds. So if anybody has attended a workshop of ours before, you've probably heard the term watershed before. But if you haven't, or if you kind of need a little bit of a refresher, a watershed is a land area over which all the water drains to a common point, and it'll bring all that sediment, dissolved materials, anything that's happening on the land into those waterways. And the boundaries of these watersheds are defined by the highest points of land around the water body. And it's really important to emphasize that the watershed is the land area. It's not just the water that's in the system, it's the entire area of land. Because again, like we're talking about with those non-point source pollution impacts, everything that's happening on the land is also going to have an impact on that waterway. So it's important to think about what we're doing on our watershed because again, everything that we're doing is going to impact our water body. And everybody lives in a watershed. So we'll just take a quick second and see if you all can identify where your watershed is. So I know that I'm in Rome, Georgia right now. So I am in the Coosa watershed up in the northwestern part of the state. If you're in Atlanta, you're probably in the Chattahoochee or Flint, possibly the Ocmulgee. I know the coast is going to be a lot harder to figure out which watershed you're in because they definitely all kind of converge together. And then we have the Savannah out on the eastern edge of the state. So yeah, you can figure out where your watershed is, feel free to comment it in the chat. We also have the 52 watersheds. So this first one over here on the left, I believe is for everybody, it's the left for me, is the 14 water basins. So these are really, really large watersheds, and you can break those up even further into the 52 watersheds throughout the state. And so this is really important to illustrate that you can kind of break down watersheds smaller and smaller, and you can have a watershed that is just the size of your backyard. If you have a little creek that's running along, you know, the back of your neighborhood or something, that, you know, it's not going to be a very big watershed, but it's going to flow into another river or into another watershed, and it can get bigger and bigger. So again, you can have it as small as your backyard or as big as the Mississippi, which drains like half of the entire nation. So, so a watershed can be as big or as small as the area that it drains. And also important to note that these waterways do run into one another. So anything that's happening at the top of the watershed is going to impact things that are happening at the bottom of the watershed and into watersheds that watershed flows into. So it's just really important to note our impacts. All right, so kind of talking a little bit more about our program specifically. So right here, this map kind of shows all of the active AdoptAstream sites across the state. I think it's even a little bit outdated. I think this is from a couple months ago. So hopefully there's a few more that are on there since then. And so hopefully this map shows that there are some AdoptAstream sites in your watershed already. But if not, then we are really excited to have you here and leading the way in your area. And so we basically show this to say that you are joining a network of volunteers all across the state and beyond. You know, we have some in North Carolina, Alabama, Florida, that are collecting and submitting data, and they come from all kinds of backgrounds, professors and students, neighborhood associations, families, you know, whatever it might be. If you're interested in getting involved and you have a waterway that you're interested in learning about, we are happy to have you. And this kind of triangle that just spun out of nowhere kind of shows the setup of our program, the structure. So as you can see, volunteers make the base of the program. You are by far the biggest group of people that we have in our program. The boots on the ground, those that are out there actually collecting the data. And you know, without such a large number of volunteers, we really would not be able to monitor anywhere close to the number of sites that are shown on the map. But we give thanks to all of our AdoptAstream volunteers for going out and collecting that data. Up next, we have our local coordinators. These are also known as trainers. So they are an integral part of the AdoptAstream program because they are, again, responsible for providing training to volunteers throughout the state, serving as a point of contact and helping you with your partnerships with local government agencies and providing, like I said, trainings and everything. We also have an advisory board of about 20 members that helps guide the program to be effective and meaningful for all of the citizen scientists that we have as part of the program. And finally, we have the teeny tiny little portion at the top, which is the state staff. You know, that would be Bailey and I, as well as Jackie and Harold at the moment. And we help support the program throughout the entire state and provide guidance where we can. I'll turn it over to Bailey.
Thanks, Cecilia. Yeah, so now that we've kind of heard about the structure of the program, can everyone hear me okay? Yes, okay, awesome, thank you. So now we've heard about the structure of the program and we know how many volunteers we have and how important and integral they are to what we do. We just want to talk about, you know, how do we get everyone on the same page? So this slide is going to show us a little bit of that. All of our volunteers are going to go through what we call QA/QC, and that's our quality assurance, quality control kind of workshops, our protocols, and that's what we're going to be talking about today. So we do have a QAP, or a quality assurance project plan, that's approved by the EPA, and that's where these protocols are coming from. So we know that we're teaching everyone the same thing, and then everyone's going to be on the same page when we're going out and we're monitoring. So to do all of that, you are required to attend a workshop, which is part of what you're doing today, going through this virtual portion. And then, like Cecilia was mentioning at the beginning, you'll also go through a field portion where you'll complete the rest of it. And then you'll pass that certification quiz, we like to say instead of test, pass that certification quiz to become an officially certified volunteer with our program. This next bullet point, only individuals are certified, simply means that since Michelle is here, I'm going to pick on Michelle because I know her. Michelle is here and she is a professor at a university. So when we certify Michelle, that means we're only certifying Michelle and not all of her students, correct? So we just want to make sure that we're doing things individually with folks, walking through all those steps together. Not to say groups can't get certified, because they absolutely can, but we just want to make sure we're taking that time with everyone individually. And then this next bullet point kind of details what I was talking about earlier. We're making sure everyone's using the same protocols when we're out there collecting that quality baseline data that Cecilia was telling us about earlier. All right, and then our last bullet point here, we're going to be talking about certified volunteers. So that's after you've attended the workshop and passed the certification. That means you can enter data onto our database, but also, as Cecilia mentioned earlier, anyone can access our 20 plus years of data that's already on the database. So it's open access, fully free and available for anyone to use, but you have to be certified to enter your data. So we kind of talked about this already as well, but to get that certification, specifically in chemical, because that's what we're talking about here today, you will have to go through this kind of virtual portion, or it could be done in person, but just talking through the background information and the context here of what you need to know in the field. So there's also a field component where we will walk through each of the steps, adding the reagents, to actually conducting the test and then understanding the test that you're going to be, or the sampling protocols that you're going to be using out in the field. And then here comes that written quiz again instead of test. And then you just have to make it 80% on that one to pass. And after you've done that, then we'll add you in our database. All right, so after you're added in, hopefully you already know why you want to monitor water quality before you get started. But if you don't, here's just some things to think about, and here's why we think it's important for us to get out and look at water quality, specifically these chemical parameters in our streams. So these water chemistry parameters give us lots of information about stream health. We're going to be talking about specifically some different 4 core parameters and then a few outside of that as well. But these things are going to tell us are organisms able to survive in the stream, and is there healthy habitat available for them, different things like that are going to show us, oh, this water is really healthy, or maybe it's not so healthy, and then we can take steps to work towards what we want our stream to actually look like and the state we want it to be in. But what's also really important about monitoring water quality is establishing a baseline. So that's something you're going to hear from us over and over again, so get used to that. But baseline data is incredibly important. And kind of like Cecilia was saying earlier too, how our program is made up of volunteers. We couldn't do any of this without you. There's so much value in citizen science because the amount of data that citizens are collecting and the baselines that we're out there establishing, no scientist could do this on their own. Even a big group of scientists would struggle to collect this baseline data. So we think it's really important. And we get really excited about folks that are out there collecting that baseline data. All right, and then we kind of talked about life in the stream already, but life is adapted to very narrow ranges of kind of what they can tolerate in the stream. So we're going to be talking about pH and dissolved oxygen and temperature. And those things can affect what organisms are able to live or not live in a stream. And then data, which we're also talking about its importance again, but it can tell us which types of pollutants may be affecting water quality. And then once we've determined that, we can kind of figure out what our next steps can be to mitigate those things. All right. So I've been talking about all these parameters, and here they are laid out for us. So we are looking at, like I said, 4 core parameters typically. There will be some exceptions for our coastal and lake folks, but we'll talk about those when we get there. But the main ones we're going to be looking at are temperature. So we look at air temperature and water temperature, as those can tell us quite a bit about what's going on. And then dissolved oxygen. So we're going to talk about all these in detail, but dissolved oxygen is how much oxygen is available in the water for organisms to use. And then pH. I think we are vaguely all familiar with that, at least in some way. How acidic is a stream, or maybe how basic. Conductivity, how many or how the amount of ions that are in the water. Clarity, so how far can you see down, how far can sunlight penetrate, and then salinity, that's specifically and only for our coastal folks. So I know we have someone from the Savannah watershed here. I don't know how close to the coast you are, but we'll touch on those things as well. And then we have in this little gray bar at the bottom, you can add some additional tests too, if that interests you. And kind of once you get a little further along in your monitoring, you can kind of see what would be of value for you to add once you get to that point. All right, and now we know what we're monitoring and why we want to monitor it. So where, when, and how often are kind of our next questions. Where, we want to identify a spot. Maybe you already have one in mind. Maybe it's in your backyard. Maybe it's in your neighborhood, on your campus, wherever it may be. You kind of have that in mind already. If you don't, that's okay too, no worries. But something you want to look for when you're kind of looking for a good place to monitor is making sure that your area is well mixed and it's flowing. So you want to make sure we can see water flowing, whether that's over rocks or whether it's a little more muddy, doesn't matter, as long as you can see some flow and that it's well mixed. So it's not sitting stagnant on the side, right? We want to make sure we can see that going there. And then once you start actually monitoring, you want to make sure we go back to that same site every time. When to monitor. So definitely want to monitor during normal flow conditions. This is very important in terms of safety. We never want to sample during high flows or after a big rain, as those can be pretty dangerous for stream access and just taking those samples. Never do anything that makes you feel unsafe. We'll touch on safety a little bit too. But that kind of relates there. But then same time of day, that's going to relate quite a bit to temperature, which I think Cecilia may be telling us a little bit about. Yeah, okay. That's going to relate to temperature and be very important in terms of consistency and again, establishing that baseline. All right, and then how often? We want to encourage folks to do this once a month, but we know you guys are our volunteers and we cherish your efforts so much. So however often you would like to go out or you can go out, it can be more frequent or less frequent, but in terms, again, for consistency, we suggest once a month. All right, now Cecilia is going to tell us about some specific parameters.
Thank you so much. All right, yes, so here are going to be our core chemical parameters. And so we're going to start off with temperature. So temperature is a measure of the thermal energy present in a substance or object. How hot or cold something is, as everyone else would generally know that as. And so we are going to be taking both air and water temperature as part of the Adopt a Stream program. Temperature is recorded in degrees Celsius, and I'm sorry, I know that I'm a Fahrenheit user just like everybody else in the US, but we're out here doing scientific measurements, so we are going to be using degrees Celsius. And we are going to be using a thermometer to take these measurements. For both of the measurements, both air and water temperature, we want to make sure that we are taking them in the shade away from direct sunlight, because if they are in the direct sunlight, then that can have an impact on the temperature. We're just trying to get the ambient conditions. And so that direct sunlight can make it seem like it's higher than it actually is. And we always want to make sure that we take air temperature before we take water temperature. The reason for this is because evaporation is a cooling process. And so if we take water temperature first and then we pull it out, the air is going to hit those little water droplets and they're going to start evaporating. And then you're going to take your air reading and it's going to seem like it's actually lower than it really is. So we're trying to be as accurate as possible. So we always want to make sure we take air first and then water. And we just take one single measurement for each parameter. The state standard, or really the threshold, the upper threshold for water temperature in the state of Georgia is 32.2 degrees Celsius or 90 degrees Fahrenheit. And there is no state standard for air temperature. So some of the things that impact water temperature. So as Bailey mentioned, we always want to make sure that we're taking samples at the same time of day, because naturally water temperature is going to vary with the time of day. You know, it's going to be the hottest at 2 o'clock or so, and it's going to be pretty cold when you get out in the morning. So we want to try to be as consistent as possible just to see the natural seasonal variation and not have it be affected by the time of day that you're going out. So some factors that can impact water temperature that can increase it are industrial or utility plant discharges. So these plants will often use stream water or water in general to cool down their systems. And then if they discharge this water into the stream before waiting for it to cool off too much or enough, then it can actually make the water temperature spike pretty high in the stream once it goes in. Also runoff from heated impervious surfaces. So things like parking lots, roofs, roads, all of those things, especially in the summer, can get really, really, really hot. And if it rains and that water flows over those impervious surfaces and enters the stream, then it can actually make the water temperature go up as well. And then removal of the tree canopy. So naturally, if you're getting rid of the shade that's covering the stream, you're going to be exposing it to a lot more direct sunlight and once again, causing it to go up in temperature. Some ways that the water temperature can be decreased are things like underground water sources or springs. So if anybody has ever swam in the blue holes on the Flint, for example, you can tell that it's very, very cold. I know that I can only stay in them for a couple minutes before shivering and running out. So that is one way that water temperature can be decreased. Snow melt, which doesn't happen a whole ton in Georgia, but sometimes we can get it, especially if you're living in the mountains. And overhanging vegetation. So kind of the opposite of that tree canopy removal. You have a lot of overhanging vegetation, you're going to have a much shadier stream, reducing direct sunlight and decreasing the temperature. So the reason why temperature is so important is because it's going to affect the feeding, respiration, and metabolism of aquatic organisms. These are all influenced by water temperature. And because life, like Bailey mentioned, adapts to a very narrow range of conditions and a very narrow range of temperatures, many aquatic organisms are very sensitive to temperature changes. And if it varies in just a couple degrees in the water, a lot of times that can mean that certain organisms are no longer able to survive there. And finally, temperature can have an impact on other chemical parameters, such as dissolved oxygen, which I will let Bailey talk about.
Yeah, so that's a nice transition for us to talk about temperature before you talked about dissolved oxygen. So dissolved oxygen, like I mentioned earlier, is the oxygen that's available in the water for living organisms, and they typically use that for respiration. So they need to breathe oxygen just like we do, but they get it from the water instead of from the air. And this relates, well, okay, the next slide's going to tell us how it relates to temperature. But we're talking about dissolved oxygen. Again, we want to bring your attention to the bold information if you're interested in getting certified. We're looking at the units here. We're going to measure in milligrams per liter or parts per million. Those are equivalent, so you can remember one or the other. They mean the same thing. All right, so then how are we going to determine dissolved oxygen? We use what's called a Winkler titration method. So you can see in this photo here, Daryl, who's one of our local coordinators, he is showing you a part of that titration process, in which you'll learn quite a bit more about in your field session. But something that's important to remember before you go into it, and just as you're kind of thinking about what this process is like, is that we take 2 samples when we're looking at dissolved oxygen. So this is something we call duplicate precision. It's kind of just something that we do because this process, there's lots of room for human error. We're adding drops in and we're doing these different things here that can impact or introduce human error into kind of this process. So we're doing 2 samples. We're doing it twice to kind of check our results, make sure they're as accurate as we can be. And so when we're looking at those 2 results, we want to make sure that they're within this pretty close range of 0.6 milligrams per liter of each other, plus or minus. So you have a little bit of a range, but we do want them to be pretty close together. And if you ever get 2 samples that are outside of that range, you want to make sure to take another sample until you get at least 2 that are within range. Again, this will make more sense once you get to that field portion, but it is bold. It is something we want you to remember. So we just wanted to drill it in here as well. All right, but now we want to kind of see what does the state standard look like. That way you have a kind of a number in your mind when you're going out into the field, you're taking your own samples, you can kind of have a measure of what we're looking for, or kind of this minimum of what we're trying to reach. So state standards for the state of Georgia for dissolved oxygen are generally no less than 4 milligrams per liter. So we want our dissolved oxygen to always be higher than that. There are a few caveats here though. So that's the most important number to remember. But if you're in a trout stream, Trout need a little bit higher. They're a little more needy than other organisms, right? So they need to have a DO, or a dissolved oxygen level, of at least 5 milligrams per liter. And then there's the South Georgia, which we love South Georgia, but it is very different and it introduces very different ecosystems, which we think are incredible. But they do kind of throw a wrench in some of these numbers. So some South Georgia streams are naturally going to have a lower dissolved oxygen level, and that's perfectly normal. There's a different type of streams, they're slower moving. It's a warmer area down there. And so we want to make sure that we keep that in mind. All right, so we want to make sure that when we're looking at this, again, we're going to talk about here. Sorry, 1 second. Sorry. Okay, so temperature and DO are something that's very important and very closely related. So like Cecilia mentioned, we want to make sure that we're testing at the same time of day, because temperature is one of those parameters that's going to impact this parameter specifically. So temperature and dissolved oxygen are inversely related. So while temperature goes up, the ability for water to hold oxygen is going to go down. So our DO levels will go down if our temperature goes up, and vice versa. The temperature goes down if it's a bit cooler outside, water's ability to hold on to that dissolved oxygen is a bit higher. So we're going to see higher dissolved oxygen levels. All right, so how is oxygen even get into the water into the first place? One of the ways is naturally by diffusion. Diffusion from the atmosphere, so kind of the air sitting right there on top of the water. We're going to see oxygen molecules naturally diffuse into the water body. But then also as a waste product of photosynthesis. So we have aquatic plants, algae, different things like that are in the water. They're going through these natural processes. And so as a waste product of photosynthesis, we're going to see oxygen introduced as well. And then one of the ways you can clearly see in this photo, you see kind of that white water across the photo, riffling, turbulent mixing or riffles. That's going to happen over rocky areas, or maybe if you have a log in the stream, something like that. We're going to see that mixing, that churning up is going to bring in oxygen to the water as well. But then oxygen can also be decreased in the water, or those levels can go down if you have higher temperatures. So again, it goes back to that inverse relationship. But then kind of how I alluded to earlier with those South Georgia streams, if you have slower moving and deep water, then the dissolved oxygen levels can actually be lower as well. And then dissolved oxygen can also decrease if you have an overload of decaying organic matter, this is typically due to excess nutrients, which we're going to talk quite a bit more about on the next couple of slides. But it's important to kind of know how oxygen is introduced into the water and how it decreases, so that we know what these levels mean when we're actually getting our results. How do we know what these changes mean, and how can we determine what to do about it? So again, it's important. Dissolved oxygen is needed in the water for organisms to breathe, respire, and live their aquatic lives. So we need to make sure that our dissolved oxygen levels are up to par. All right, so our next few slides here are going to be talking about eutrophication. Sounds like a big word if you're not familiar with it, but really it's a simple and natural process. If a body of water becomes excessively enriched with nutrients, this is something that humans can kind of speed up that process of, as we're known to do with a few different things in nature. But if a body of water becomes excessively enriched with nutrients, then we're going to see a few things happen. We're going to see an algal bloom, which is typically brought on with these different types of nutrients in the water. So these algae like to feed on these nutrients that are being introduced, which are typically nitrates and phosphates. So you see those here on the screen. Those can be anything introduced by different fertilizers, maybe industrial waste. Sewage is a main contributor in a lot of, especially urban areas. We also have animal waste and things like that as well. So if those things are getting into the water in high amounts, and then we have these algae that are really excited by that because they love those nutrients. They want to feed on those. It's lots of good food for them. So then they're feeding on all of those, but then we have an eventual decrease. Yeah, thanks, Cecilia. I was getting there. I was like, oh man, I need the graphic. She's got me. So we can see this number one here, excess nutrients comes into the water body. Number two, rapid plant growth occurs because again, they're feeding on all that good stuff, right? So when that's happening, they are photosynthesizing because they're getting, food. They're photosynthesizing to produce that food. And then, so we're going to see a spike in DO, right? But then here's where the bad part comes in. These algal blooms will block the sunlight, and then the plants that are kind of down there on the bottom will die because they're not be able to access that sunlight. The decomposers are going to start feeding on those dying plants and dying organic materials, and then we're going to see dissolved oxygen levels go way down compared to what they were. Excuse me. So then once we're at that point, oxygen levels can get so low that aquatic life can no longer be supported. So a lot of times this is where we see fish kills come in. So I know that's kind of a lengthy explanation, but what we need to remember about this process is if we have excess nutrients coming into the water body, we're going to see an initial spike, but what's really going to happen is a big decrease in dissolved oxygen levels, which can be really harmful for the life in the stream.
Great, thanks Bailey for that awesome explanation. So up next, our next parameter is pH. So this is a measure of the hydrogen ions that are present in a substance, but more simply, it's how acidic or basic a substance or waterway is. And so there are actually no units for pH. It is measured on a scale from 0 to 14, with 7 kind of being our neutral number. If it's less than 7, then you're going to be in the acidic range. And if it's higher than 7, then you're going to be in the basic or alkaline range. And so the way that we measure this is with a color comparator. So you can kind of see the thing that's being held up in that picture right there. So essentially you'll add some reagent into your water sample and you'll plop it into these little color comparators and try and see kind of which color it most closely aligns with. And you're going to take 2 samples, again, for duplicate precision, like Bailey talked about with dissolved oxygen. Goodness. You're going to take 2 samples for duplicate precision. And these samples need to be within plus or minus 0.25 of one another. And you can see on the color comparator here that the numbers are in 0.5, so 7 to 7.5, 8, 8.5. And so you can actually estimate in between those numbers. If you think that, oh, it's a shade, lighter or a shade darker than those colors, you can estimate in between. But your 2 samples need to be within 0.25 of one another. And if they're not, then you need to go out and take another sample until you do get 2 that are within that range once again. So the state standard of pH is between 6 and 8.5, and this is actually a pretty wide range, but this is the state standard for the majority of the state. But once again, South Georgia streams are a little sneaky, and they can actually get down to as low as 3.5. That is very acidic. We were out on the Okefenokee recently, and we were getting some 3s and 4s-ish, really, so it's very acidic. And that's due to a very natural process in the region. There's natural vegetation that when it gets in the water, it introduces tannins, which are acids. And so that can cause the pH to decrease in those systems, but it's totally natural. And all of the organisms that are living in those systems are very adapted to live in very low pH levels. So it's all good. And also along the coast, with higher salinity, you're going to see increases in pH. So the pH is going to get more basic. So some of the things that can impact pH, pH should remain relatively consistent. But if you do notice a deviation, then that can mean that something a little fishy is happening in your system. So pH can be increased by photosynthesis. This is because photosynthesis is naturally removing carbon dioxide from the water, and carbon dioxide is actually mildly acidic, fun fact. So if you are seeing really high levels of photosynthesis, for example, like what Bailey talked about with eutrophication, you may see a little bit of a spike in pH. Additionally, calcium carbonate, which is lime, is naturally released in areas that have limestone bedrock. And so that can also increase the pH. pH can be decreased by respiration, which is the exact opposite process of photosynthesis. So it's adding carbon dioxide into the water. So it's going to make it a little bit more acidic. Tannins, which is what I talked about in the South Georgia streams. And you can actually see in this photo that is tannic water. It's got that kind of tea color. And that is really what it looks like. If you take a sample out, it's going to be kind of like a light brown color. Again, totally natural. Something that is not totally natural is mine drainage, which is another way that pH can be decreased. And so that can actually have pretty harmful impacts. And there's a few other ways as well, like, climate change is another way, because again, it's introducing carbon dioxide into your system, which is making it more acidic. And the reason why this is so important is because a small change in pH can represent a very large change in hydrogen ions that are present. And again, like those organisms that are living in your system are very adapted to a very narrow range of pH. So even a small change can mean that they're not going to be able to survive in that system anymore. And also pH, different pHs can impact the solubility of nutrients and heavy metals, which can be toxic in a system. And here's just a cool little graphic that we wanted to show off that kind of showcases the different pHs of different kind of household substances, that you might see. So you can see that, Coca-Cola has a pH of 3, but if you go down just by 2, then you're in the hydrochloric acid range. So you can see, like, you can drink a Coke and be okay, but I would not recommend drinking hydrochloric acid. I think it's going to be pretty bad for you. So again, that just kind of showcases how just that little change in pH can have a really, really big impact on the system.
Awesome. Now we're going to talk about conductivity. So we do want to draw your attention that this is the only definition that is bolded in the PowerPoint. So yeah, conductivity is a measure of a substance's ability to pass an electrical current, or it means the same thing to say that it also indicates the presence of ions in that substance. So conductivity can really tell us a lot in streams, but something that's really important to remember here again, because it's in bold, are those units. So we measure using a little meter, and that meter is going to measure in the standard unit of microsiemens per centimeter. So you don't have to do any calculations to figure out what that means. That little meter will do that for you. But those units are important to remember. All right, so actually taking your measurement might be hard to see in the photo, but there's a little green meter that our friend Kristen here is holding and taking a conductivity reading with. Since we're using these meters, we're kind of eliminating as much as possible human error from the equation. So we're only going to be taking a single reading with these. And the reason we're able to do that is because we calibrate our conductivity meter before we go out and take that sample. So your conductivity meter should be calibrated within 24 hours of you taking a sample. So it doesn't need to be, exactly 24 hours before you go and take that sample or anything like that. Just within a 24-hour period of when you're going out, you should calibrate that meter. And your trainer will probably talk you through that process once you actually get there, but it should be easy peasy, no problem. All right, so state standards. So we've been telling you numbers throughout this time to kind of give you an idea of what things mean and kind of what we expect to see in the state of Georgia. We're not going to be that helpful with conductivity. So there are actually no state or regulatory levels for conductivity in the state of Georgia. And that's because the state of Georgia is very unique, and we have lots of different types of ecosystems and regions within Georgia where the geology differs so much. And we'll get to why geology is important here on the next slide. But we have such a range here in Georgia. And so we just kind of have a general range of what we tend to see. So we have 50 to 1500. Now that's a massive, massive range, right? So if you're not somewhere within there, there might be an issue. we definitely want to try to stay within that. But kind of how Cecilia was talking about that, that 90 degrees Fahrenheit being our cutoff for temperature, this is kind of similar with conductivity. We generally do not want to see anything higher than 1500 microsiemens per centimeter reading, because something is likely wrong. We don't usually see readings that high. But it's all about determining your baseline. That's especially important with conductivity. So we can, we're taking consistent readings every month, and you're going to find out what's normal for your stream very quickly. And so that's why it's very important to go out every month and do that consistent monitoring so we can get that baseline, figure out what's normal. So determining what's normal, something that determines that in the state of Georgia is geology. Geology determines what our baseline conductivity levels are going to be. So obviously we have some different types of geology within the state of Georgia, so we're going to expect to see some different results. If you are in the northern part of Georgia, you are probably familiar with Stone Mountain. That's just a huge piece of granite right up in our area. So we have some granite bedrock that's naturally found in North Georgia. But sometimes even into West Georgia a little bit, but like especially Southwest Georgia, we're going to see lots of limestone, and that limestone bedrock is making up a lot of the geology there. Especially, and then down in South Georgia too, we're going to see lots of that famous clay that was talked so much about in songs and different things. We have that red Georgia clay that's also going to be impacting the geology. So these, and salinity, sorry, here at the other side too. So on the coast, having salinity or that seawater there is going to increase your conductivity. So if we're thinking about what conductivity means, and we think about it in terms of the amount of ions that are in the water, we're going to expect conductivity to be quite a bit higher when we have, say, increased salinity. So if we have salts in the water, that's the type of ion, right? So we're going to see a much higher conductivity level. The same can be said for limestone or areas with limestone bedrock and clay. That type of substance is going to degrade or give off ions a lot more easily than, say, granite would. So we're going to see in areas with limestone bedrock a much higher conductivity naturally than we would see in an area with granite bedrock. Now, these things can be changed. These are the natural factors, and that's going to determine our baseline. But there are factors outside of that, human factors, of course, that are going to impact our conductivity levels. So mining operations, that's what Cecilia mentioned as one of the factors for pH as well. But if you have mining operations, people are trying to get these precious metals and things out of the earth. That's naturally going to release ions as well into the water bodies. So we could see a spike there. Agriculture, so if you have agricultural runoff. We kind of talked about those nitrates and phosphates getting into the water. Those things are definitely going to increase your conductivity as well, because nitrates, phosphates, those things are ions. We're going to see that spike. Sewage effluent, which is what you see in this lovely photo here. Ugh, gross, right? So sewage effluent is definitely going to introduce some things that are not natural into our water body that are again going to introduce ions and give us a spike in conductivity. And then urban runoff, there's just a whole slew of things that we don't even know that's getting into our water when that happens. These big rain events can introduce quite a few different substances that are again going to increase that conductivity. So it's important. Conductivity is one of the easiest measurements to take, very simple, but it's going to tell us so much information, especially once we establish that baseline. Because it's so quick and easy to take that sample, you can kind of follow samples, or maybe follow problems upstream until you find what's wrong. So if you have an abnormally high result after you've been monitoring for a few months, then you can kind of go look around you, maybe take a smell around you. Maybe you smell some, hopefully not, some sewage effluent, or maybe things that are going wrong in your stream. And conductivity measurements are a pretty easy way to kind of check in and do a quick and easy result. But however, there is a caveat here. So we have, these results, we can see they're high, they're low, they're normal, but we don't know just from those numbers what types of ions or potential pollutants that are in the water body. That's something that's going to require further testing and further investigation to figure out what those things are. But again, this is a good indicator or a good push in the right direction of figuring those things out.
All right, so next up, we're going to talk about salinity. So this is going to be for coastal volunteers only. If you are monitoring a freshwater system, which does make up the majority of our program, then you won't be dealing with salinity at all. But if you are on the coast or in some of those brackish areas, those estuaries, then you will be using salinity because it kind of, in a way, takes the place of conductivity, because you cannot really take conductivity measurements when you're in brackish or saltwater conditions, because it's just going to show a really, really high conductivity regardless, because those salts that are in the water, like Bailey mentioned, are ions. And so it's not really going to tell you a whole lot about what's in the system. It's just going to tell you that it's really salty. So salinity is kind of taking the place of conductivity for our coastal volunteers, and it is the measure of the amount of dissolved salts that are in the water. And so salinity is measured in parts per thousand. So how many parts of salt per 1,000 parts of water, essentially. And it is measured using a refractometer, which is what these ladies are using in the picture here. Once again, like with pH and with dissolved oxygen, we take 2 samples for duplicate precision once again, and those samples must be within plus or minus 1 part per thousand of each other. And again, if they are not, just like with all of the other ones, you do need to take another sample until 2 are within that range. Now, just like with the conductivity meter, refractometers must be calibrated within 24 hours prior to each monitoring event. And you can just use deionized water to do so, which would have a salinity of 0. It gives you kind of that known baseline condition for salinity. Now, there is no state standard for salinity in the state of Georgia because it's really going to be super dependent on the tidal stage and whatever freshwater inputs are coming into the system. And so, yeah, there's not really going to be any sort of state standard because it is going to fluctuate pretty consistently. But just as kind of a reference, the salinity of seawater, like pure seawater, if you're out in the ocean, is going to be around 35 parts per thousand. So generally in brackish conditions, you're going to see somewhere between, I think it's 0.5 to 35 parts per thousand. That's generally what you're going to consider as brackish water conditions. It's a pretty big range, but. All right, so some factors that impact salinity. Naturally, the tides. So as the tides are moving into the estuaries, into the land a little bit further, then they're going to bring higher salinity levels into the systems naturally. Also, water temperature is going to have an impact on salinity. So it's in the exact same way that it has an impact on dissolved oxygen. So warmer water is not going to be able to hold as much salt in it, essentially, as much dissolved material as cold water is going to, because it is less dense. So as the water temperature increases, the salinity is going to decrease, and vice versa. Some human factors that can impact salinity. So we have saltwater intrusion. So this happens during periods of drought, or if there is excessive pumping of groundwater sources, that's going to reduce the freshwater inputs into estuary systems, essentially. And so if you have fewer freshwater inputs, you're basically going to have more saltwater inputs coming the opposite direction. So that saltwater is going to work its way a little bit further up into the mainland than it normally would. And so you're going to see higher levels of salinity in areas that you normally would see maybe more fresher conditions. And then we also have sea level rise, which is an impact of climate change. And that's essentially meaning that the ocean water is getting a little bit higher. And so it's going to move further inland and start kind of overtaking some of those freshwater and brackish systems and making them have higher salinity levels as well. And so the reason why this is important is, just like with basically all of our parameters, the majority of aquatic organisms are adapted to a certain level of salinity. There are some that are adapted to, deal with fluctuating changes. Like blue crabs are pretty good at it. The Spartina grass that's in here can adapt to really, really saline conditions. But most organisms are adapted to just a very specific level of salinity. And so again, if you see salinity changes, then those organisms may not be able to survive in those conditions anymore. So next up, we have water clarity. So again, this is not going to apply to our stream volunteers. This is only going to apply to our coastal and our lake volunteers, because you can't really have flowing water if you're doing this test, and we'll kind of get into that in just a second. But water clarity is essentially the transparency or clearness of the water, so how far you can see down into the water body. So water clarity is measured in centimeters of depth. Oh my goodness, I could not say that word. It did not come out. Centimeters of depth. Gosh, it's really not coming out. Anyway, you know what I'm saying. That's essentially how far you can see below the water surface. And so what we use is called a Secchi disc. So it's kind of this black and white disc that you lower into the water and mark essentially how the deepest point where you can still see the disc. So you'll lower it down until you can't see it, and then lower it ever so slightly until it's just visible, and that's going to be your depth. And so you'll pull it out and measure the string distance, and that's going to be your measurement for water clarity. And so you can see why we can't really have a flowing system if we're doing this, because then your disc is going to be all the way over there, and it's not going to make any sense. So really just for those lake and coastal volunteers. Once again, we need to take 2 samples for duplicate precision, and those samples must be within 10 centimeters of one another. And again, if they are not, then you need to take another sample until 2 are within that range. Once again, we do not really have any state standard for water clarity in Georgia. And you can also test for turbidity. It's another way to test for water clarity, but it is a little bit more of a cost prohibitive test, because you need something called a turbidimeter and a lot of very specific equipment for that. All right, so some of the things that can impact water clarity, some natural factors. So rainfall is naturally going to disrupt the surface. It's going to carry some soil in with it. When it's flowing over the land, it'll bring some, sediment and whatever might be on the land surface into the waterway. Also tidal stage. So in areas that are impacted by tides, the water flow that's coming in or coming out can be strong enough to resuspend sediments that are on the bottom. So you can actually have a little bit of turnover, which can decrease the water clarity. And then algae growth, which actually can be very natural. So algae does not necessarily have to be big algae blooms, whatever, that are being caused by eutrophication. They are present in a lot of natural systems, and they can, still form a little bit of a layer on the surface, which can impact water clarity. Now we do see under human factors, eutrophication is an example, and this is when you're going to see those huge blooms that you're going to get from algae, as well as just a bunch of decaying organic matter. And so once again, that's going to reduce the water clarity. Also development. So if you have construction projects and you get some erosion, especially if it's combined with rainfall, all of that sediment, is going to flow into the stream and kind of, create that muddy sort of look, which I know I often see on the Chattahoochee near me after a big rainfall, especially with all that red Georgia clay coming into the river. And finally, dredging operations, which once again will stir up the sediment that is in the water. And so the reason why this is important is because a number of organisms, and specifically aquatic plants or things that use, or algae or anything that is utilizing photosynthesis for food, rely on sunlight getting through the water and hitting them so that they can, conduct photosynthesis. And so if you have really low water clarity, then it's going to limit the amount of sunlight that's going to be able to penetrate the water and for them to use for photosynthesis. And additionally, if you have reduced water clarity due to suspended particles, so from things like erosion, from that clay that's suspended in there from construction operations, then those suspended sediments can actually get into the gills of fish and little macroinvertebrates and things like that, and can actually suffocate them. And for mussels and clams and things like that, it can also disturb their ability to feed, because they're going to be filter feeding. And so if they're getting all clogged up with all the sediment that's in the water, then they're going to have a lot tougher time getting the food that they need.
All right, so we've talked through all of our parameters now. So you made it, everyone. We're almost to the end here. I put a few comments in the chat, but I just wanted to briefly mention that before we left off of our parameters, that if you're a coastal volunteer, tidal stage takes precedence over time of day. I should have said that at the very beginning, but it slipped my mind. Tidal stage does take precedence over time, specific time during the day when you're actually taking these samples, because that's going to impact your results much more than sampling, at 8:30 versus 9:30. So generally the same time of day, morning at the same outgoing tide or something like that is what you're going to want to aim for when you're doing this type of monitoring. And then also, I put a little note in there too, but if you're taking a water clarity sample, make sure to take your sunglasses off, because that's going to impact. Sometimes you have those fancy polarized glasses that will impact how far you can see. Anyway, just a couple things to throw in before we get into kit maintenance and how to take care of your reagents that you're actually taking these samples or using to take these samples. So when you get a chemical kit, you want to make sure to store it in a cool, dark place, just to make sure that we're not exposing our reagents and our chemicals to high temperatures or things that can impact their functionality. We want to make sure that we note the expiration dates. So you can see we have an expired one here on the slide. I want to make sure to note those dates when they expire so that they can be replaced before they do. And then we want to keep an eye on them too to make sure they're not contaminated. And you'll be able to learn a little bit more about that in your field session. But just making sure that you're keeping the lids on the right containers. We're not sucking up any liquid into any of them or anything like that. We just want to be careful that our reagents are functioning as they should be, so they get the most accurate results as possible when we're doing these tests. All right, so after you've done all these tests, you are going to have some wastewater, because we don't want to do all these tests with all these reagents and then just dump that vial that's full of water and chemicals back into the water. We want to make sure we put that to the side in a clearly labeled waste container, and we take it home and dispose of it properly. So if you're on a municipal system, then you are perfectly fine to just pour that stuff right down the drain, flush it down with some water. Your municipal system, trust us, we work with a lot of those. They say there's way worse stuff that comes through than these diluted chemicals. So if you're just getting rid of your wastewater from your monthly sampling event, you should be fine, as long as and only if you're on a municipal system. If you're on a septic system, I'm going to be sure not to mess with those lovely critters that are taking care of our septic tanks, right? So we don't want to pour any of these reagents down our drains if we're on septic. You can take them to a municipal facility, your public library, or the McDonald's in town. They'll never know. It's fine. Or you can actually solidify your waste. So you can use some kitty litter and then seal it in a bag or a jar or something like that and toss it out. So that's another way, if you're on septic, to get rid of that waste. If you ever need reagents, you can always contact us at the state office or your local coordinator, who hopefully you'll be in touch with after this virtual session with us. Because we are happy to provide equipment, and typically so are your local coordinators. But if you have any questions about that, please feel free to contact us. All right, so after you've gone out and you're done your results, you've gotten your results by taking all of your measurements, there's going to be some things maybe that you need to do besides just putting in your results on our database. If you find any results that are outside of those state standard ranges. So remember, we have a state standard for DO, we have a state standard for pH, or if you see anything that deviates very strongly from your baseline levels, and that goes for anything, not just conductivity, not just DO, any of those that are deviating very strongly from your baseline, we want to make sure that we are investigating our site to identify possible sources of what's causing those issues, those deviations, or those results that are outside of our state standard range. So that can look like, usually you're doing these tests right at your stream site, or maybe your lake site, or whatever it may be. You're right there and you're getting those results pretty quickly. And then if that is the case, you're right there on site doing it. You can kind of walk upstream just a little bit, take a look around, maybe take a sniff around, see if you're finding anything, right, that looks abnormal, that smells abnormal, things that you're not used to seeing there that could potentially be causing some issues. So if you find something, maybe you find some sewage spilling into the stream, or maybe you find some cows wading through the stream, something like that that's not normally there, that's causing a difference in what you're used to seeing. That means that you found a source. Hooray, that's great news, because then you know what to do after that, right? You can notify your local coordinator with Adopt a Stream, and or what we typically say, and your local water authority. So those people know what to do when we encounter issues like that. If you find a sewer spill, if you find things that are abnormal, those folks are able to come out and mitigate those issues right away, especially if you find a source. So if you don't find a source, you've looked around, you don't find anything that seems abnormal, just take another result, another sample to see if you get the same result. And then generally we like to advise people to kind of check upstream of themselves to see if those results yield anything different. If they're still within that range and you kind of have those still similar results, but you're not seeing anything, then maybe just come back and try again and see if you get anything different. If they're outside of that range still, you definitely want to notify someone. But if you've got to go upstream and you get results that are maybe normal, it's just a fluke maybe. So just monitor as usual. I hope that makes sense. But we want to just make sure that you realize if you find a source, definitely notify someone. If you don't find a source, but things are still abnormal, even after you've tested again, notify someone. Safety. I did say we'd be back to this, right? So safety is very important to Adopt a Stream, because again, our volunteers, they're our whole program. You guys are a part of why we do what we do. We're the whole part of what we do. So we want to make sure that everyone is being as safe as possible when they're going out. We tell folks to try not to sample alone. Everything's more fun when you take a friend, right? So take a friend with you, even if they're not Adopt a Stream trained, just have them go out, have a picnic at the stream with you or something like that. If you don't take a buddy, make sure you text, call someone, let someone know where you're going, because you really never know what's going to happen. Maybe a rock is more slippery today than it has been in the past 3 years. You just don't know. So we want to make sure that we're as careful as possible. Again, I think I alluded to this in the beginning, but we want to make sure we never sample during high flows or after a very heavy rain event. That can make stream access or site access pretty dangerous. So we want to make sure that we're not doing that. And then, very important one, again, make sure you obtain permission if you're going to sample on private property. We do not encourage sneaking. We want to make sure we have permission to access any property that we're on. So be sure to do that before you go out. And then wear your proper protection equipment when sampling. So we suggest boots, gloves. I know a lot of our photos in here don't have gloves on, but that's because we're doing them in workshops and we're just practicing. When we're actually out in the field, we do suggest you just wear that proper protective equipment, again, just to be safe. And this is not an exhaustive list of all the safety things. We just want to make sure if you ever feel uncomfortable, don't go out. Getting that monthly result is never more important than your safety. Yeah, even though we talk about data and we love it, we think it's very important, it's not more important than your safety. So again, just be careful when you're going out and keep these things in mind. All right, so once you're certified, you've done all of these things, right? of you've been through, you sat with us, with me and Cecilia, and you you've sat through this presentation. Then you go and get your field certification, you take that quiz, and now you're certified. So you will get an account on our online database. You'll be sent an email that's going to tell you how to sign in and all that good stuff, and you'll have a profile on our database then. And after you're certified, you'll be able to submit data, because remember, only certified volunteers can submit data, although anyone can look at it and access it. Your certification is valid for 1 year. So after that 1 year, you will need to get recertified with our program. That doesn't necessarily mean you have to sit through this whole workshop process again. It just means that we just want to check in with you, make sure that those protocols are still being upheld. Everyone remembers those processes of how we're doing these tests. And then we're just going to get you to take that test again, or quiz, excuse me, the quiz again, so that we're all on the same page. This is just maintaining our QA/QC. Remember that quality assurance, quality control protocol. All right. Yeah, so we want to keep those things in mind. All right. So we're talking about data. After you've collected it, you're certified, you have an account, then you're going to input the data that you're collecting into our database. We get questions all the time. What are those data actually used for? What does it mean? Is it just sitting there? Well, no, and we encourage you to not let it sit there either. You can kind of take this data into your own hands and do as many things as you want to with it. We encourage anyone to access, download, and analyze or display in however they'd like to. But here are some ways that we use, and we know other folks use your data as well. So again, maybe you're tired of hearing it, but they do establish baseline conditions for water bodies across the state, which again is incredibly important for some areas that are pretty remote that maybe folks don't normally get to. Like Cecilia was saying, we were lucky enough to go and sample on the Okefenokee Swamp in the state of Georgia a few weeks ago. And I wouldn't say that the average Joe typically goes in there, every week. So it's pretty neat if you're in a area that not a lot of people live or not a lot of people visit, you can kind of establish baseline for that area, which is incredible and has lots of value to science and to our state organizations. They can also help you discover and report water quality issues. So kind of how I was talking you through, maybe walking upstream, finding what's wrong. Those things are very important because it gives us the power as citizens, as volunteers, to take things into our own hands and make sure these problems are getting resolved. So you know what the issue is, and you know what caused it. A lot of times that's going to help speed up the process of fixing it so much. And then educating your community, which has such power. These data can tell us a lot about different types of things that are going on in our water and kind of what this water means to communities. So that can be really powerful as well. And then we've got, no, you're fine, you're good. And then if anyone's familiar with the 303D or the 305B list, if you're not, totally okay. If you are, great as well. But there's lots of information on the Environmental Protection Division website that can give you some more information if you're interested. But these lists basically are going to tell us if these water bodies are meeting certain requirements. They're recreational requirements most of the time. So maybe if you've heard of like, this water body is suitable for fishing or fishing and swimming, different things like that. So we want to make sure that our water bodies are suitable to recreate in. We don't want to get in any type of water body that's going to be harmful to us, our family, our pets, anything like that. So the Georgia AdoptAStream data can help inform the status of these streams and can kind of help serve as a justification for altering these stream statuses. Again, if you'd like more information, we're happy to provide that. But these are just some ways that your data are used, but the possibilities are endless, and we encourage you to look at it, check it out on your own.
All right, we are so close to done, I promise. And we're gonna just run through these next slides and wrap it up. And so we're just gonna talk you through our database and how to access it and log in. So once you get certified, then you will get an account, and this is the sign-in page. You will need to log in In order to access the data submission forms, because once again, only certified volunteers can submit data to the database. And you can find all of this information as well on the Adopt a Stream website. We have a whole volunteer FAQs page, which can help you guide you through the process. But once you log in, you will have access to the data submission form. So this is a screenshot from the website. It's under data entry, data submission form on the top. Again, you must be logged in order to access the data form with an up-to-date certification. And this is where you're going to enter all types of data that you will collect. And so now we will go through and show you how. Oh, well, first off, we'll show you, this is the paper data form, the chemical monitoring form. And so it's very similar to the online forms. So it has all of your site information, your group name, group ID, stream name, participants, all that good stuff, and as well as observations. So like we talked about, the O in ADOPT stands for observations. It's a very, very important part of our program. And like I said, every single time you go out, regardless of what program you're involved in, you will fill out this observations part of the data sheet. And so like I said, like water clarity, water color, water surface, water odor, all of those kinds of things can tell you a whole lot about your stream. And so this is the chemical part of the paper data form. So this is where you'll fill out your results for air temperature, water temperature, pH, all of those core parameters that we talked about already. And it also has some spots for additional tests if you happen to do like Secchi depth with it. Depth, oh my goodness, I can't say that word. It's not working tonight. Anyway, if you have like nitrates or phosphates that you're testing for, you can fill that stuff out here as well as any comments, additional observations that you may be seeing as well. And a note on here is to remember to submit as soon as possible to the online database, especially if you notice anything that's a little funky. We definitely want that inputted as soon as possible in order to catch those issues, because all of your submissions are going to go to both the state office and your local coordinator. And so just maintaining that communication and making sure to keep your data submissions up to date. And also, you're not going to want to submit, 12 data sheets worth of data at the end of the year. It's not going to be nearly as fun. All right, so just going through quickly on how to submit data. We are currently working on updating our database, so this may change kind of soon, but for the time being, this is how you'll go about it. So you'll fill out all of your observations, all of your site information, and then you'll go to the bottom, scroll all the way to the bottom once you're finished, and click submit. And then once you've clicked submit, you will navigate to the chemical tab, and you will start filling out all of your chemical data as well. And then once you're done with that, you will hit submit and you will be all done. If you're doing bacterial macroinvertebrate, you can also just go onto those tabs, or the stream habitat survey, any of that stuff. And you'll just have to click submit after each form to make sure that it is entered into the database. All right, so that's basically it, an hour and a half later, and we've told you all that we have to tell you about chemical monitoring. We do urge you to stay connected with us. So, here, this is the state staff. We have Harold, who's our manager, Bailey and I, who are the state coordinators, and Jackie Encinas, who is our outreach specialist. We are all happy to talk to you if you have any questions, concerns, or just want to get involved. This is our email that's on here. We can also put it in the chat. We also have our website. We are on social media. We have a Facebook account and an Instagram account. So be sure to follow us because we post some fun stuff on there. And then just our mailing address and our phone numbers. So yeah, be sure to connect with us. Again, that is all that we have for you. So I will go ahead and stop sharing and use the 2 minutes that we have remaining here to answer a few questions. So I think we have a couple in the chat.
Yeah, I've been replying to a few in there.
Awesome.
Sorry. I hope those answers help. If they don't, please feel free to unmute and elaborate, or I can speak, or Cecilia can speak, or Jackie can speak to those things a little bit more. I saw Bob, you did ask, why did you decide to add eutrophication to the course? Is this a significant issue in the state? I can speak to that a little bit. So the reason we added that was because if you remember, which I'm sure Bob you do because you've taken our test a few times, is that we ask our trainers and our volunteers to know what effect excess nutrients has on dissolved oxygen levels in a water body. And it wasn't really explained in the old PowerPoint why dissolved oxygen levels went way down if you had excess nutrients. So we just wanted to put some more information in there about it. It's not necessary for someone to be able to explain the whole eutrophication process to be a part of our program, not at all. But we just wanted to provide some context for that.
If anybody has any further questions, feel free to unmute and just shout it out, or throw it in the chat like the other people here have done. So however you want. Also, if you are interested in doing the field portion of the certification, please let us know. So you can either reach out to us. I can put, once again, I can put our email in the chat. Yeah, you did. Okay, cool. Bailey's going to throw the email in the chat. So feel free to email us after this. Let us know that you attended and that you're interested in doing the field portion, and we will work on connecting you with your local coordinator to get that done.
Thank you everyone for joining us tonight and sitting through Cecilia and I talking through everything. So we're so excited to share this new PowerPoint with everyone. Share some of the updates that we've been cooking up. So hopefully you've enjoyed them. And I think like Cecilia said kind of before we got started, our bacterial and our intro PowerPoint recordings can be found on our YouTube, where this one will be added. So feel free to just check those out. We have a few different video resources there. But yeah, we're excited to be working on updates and sharing them with you. So let us know if you have any feedback, questions, any of that good stuff. We're excited to hear it.
All right. Well, if there are no more questions, I think we'll go ahead and wrap it up. So like Bailey said, thanks so much for joining us tonight. And please feel free to reach out to us if you have any questions or would like to get connected to your local coordinator. All right. Good night, everybody. Thanks so much.
Thank you. Good night.